Thank you. It's a pleasure to be here. We could talk about the biopirates, but I think we'll leave that for another time. So I congratulate you on this really excellent symposium and rigorous and open science. I think it's wonderful. And today I want to explore with you, can we advance this, or what's the issue here? Which way?
It's this one, and I think you have to point it. This way.
Okay, good. Now, so today I want to share with you, explore with you gendered innovations. Usually I take people directly to the website so you can see it as a resource, but there seem to be technical issues, so we're just using slides. Gendered Innovations was produced through a large international collaboration involving the European Commission, the US National Science Foundation, and Stanford University. And we're now expanding, so we networked scientists across Europe, the US, Canada, and we're now expanding into South Korea, Taiwan, and Japan. Gendered Innovations has brought together over 80 basic scientists and gender experts in a series of collaborative workshops. So you may say, how is this relevant to my work? So first of all, if you want to create health science that works for humankind, I'm not using the word that was used last night, mankind, I'm using humankind, which is the inclusive term, and you want medicine to work for both men and women, people of various gender diversities, and if you want personalized medicine, you have to integrate sex and gender analysis into your research. So that's number one. Number two, if you're looking for funding agencies are increasingly asking for sex and gender analysis in the research in their call for proposals. Of course, the European Commission has done this in a marvelous way in their Horizon 2020. The US NIH in 2016 now requires for all public funded science that sex be considered as a biological variable, very important. And if you want to publish, this is also part of the, not quite requirements, but guidelines by journals. Last December, Lancet adopted sex and gender analysis, so you have to at least know what the sex of your animals, cells, and humans might be. And also last December, the International Committee of Medical Journal Editors adopted these as guidelines. So important reasons to incorporate this into your research. However, this is rarely taught at universities. Charité, I think, really has done wonderful things in integrating sex and gender, the results of sex and gender research, into its curriculum. We at Stanford have not managed this yet, and I don't know of many universities that have, so congratulations to Charité, very important for the future generations. So innovation is about integrating sex and gender analysis into the design of research. The operative question is, how can we harness the creative power of sex and gender analysis for discovery? Does considering gender add a valuable dimension to research? Does it take research in new directions? So, just a bit of background. This machine doesn't like me very much. This way?
Yeah, and then on the right there. Uh-oh.
Here we go, here we go. Well, what did you do then?
Yeah, but you're right, it doesn't like us.
Usually we need new batteries. It's really simple.
Okay. For some reason now it works.
Okay, well. So a bit of background, just so everybody's on the same page here. Governments, universities have taken 3 basic strategies to gender equality over the past several decades. The first is fix the numbers of women, focusing on increasing the number of women participating in science and engineering. The second is fix the institutions which promotes gender equality in careers through structural change in research organizations. And the third is fix the knowledge or gendered innovations, which stimulates excellence in research by integrating sex and gender analysis into the work. So this talk today, I'm going to speak only about the third strategic approach. It's the newest and hottest area and the most important for the future of science and innovation, and it's what gendered innovation is all about. So let's dive in. If we could, we would dive right, there we go. Okay, so doing research wrong costs lives and money. For example, 10 drugs were recently withdrawn from the US market because of life-threatening health effects, and 8 of those posed greater threats for women. Not only did these drugs cost billions of dollars to develop, but when they fail, they cause human death and suffering. We can't afford to get the research wrong. But doing research right saves lives and money. An analysis of the US Women's Health Initiative hormone therapy trial, which was a large government-funded trial done in the 1990s, found that for every dollar spent, $140 were returned to the public in health care that was not required. The study also saved lives. There were 76,000 fewer cases of cardiovascular disease, fewer breast cancers, and more quality-adjusted life years. And while most of the results were positive, there were more osteoporotic fractures. I really like metrics like this and would like to develop more. So it's crucially important to get the research right from the very beginning, and this is the goal of the Gendered Innovations Project. On our website, you find state-of-the-art methods for sex and gender analysis. If you did not learn this in school, you can go through our 12 methods, which are basically just questions that researchers pose as they design the research from the very beginning. If you set a bad platform, it's much harder to change it later on. So we develop methods, and we also provide case studies, which are concrete examples of how gender analysis leads to discovery and innovation. And we have 26 case studies there in basic science, health and medicine, engineering, and also in environment. So the first example, the first case study I would like to look at comes from cells and tissues and looks specifically at stem cell therapies. So let's go back to why 10 drugs were withdrawn from the market. There are many reasons why drugs fail, and fail more often for women. One reason is that in most research is still done in males, whether humans, animals, or cells or tissues. And this study, which was done in 2011, but is still valid, was done by some of our colleagues at Berkeley, and it shows the sex of the animals used in research. The blue here indicates that males are more often used than females in all areas except reproduction and immunology. But what I'm interested in is this gray area over here, and that is where the sex of the animal was not recorded at all. This is research money wasted. You might as well throw it out the window. You can't necessarily reproduce this study because you don't know the sex of the animal. You can't take the data and power up another study because you don't know the sex of the animal. You have to know the sex of the animal. A similar study was also done in 2011 at Mayo Clinic on cells and tissues. And here, it's on the other side, but you see only gray area. The sex of the cell is almost never reported, and again, this is money wasted. So let's then look at our stem cell case study. Why might the sex of the cell be relevant? So research shows that there are sex differences in the therapeutic capacity of stem cells. And this slide simply shows stem cells taken from muscle tissue and indicates that female cells are more regenerative or more active than male cells. Yet very few researchers consider the sex of the cell, and that can lead to failure. An international team from Norway and Australia worked with stem cells in mice, and they were appropriately using male and female mice, but then they used all female stem cells, and this was an arbitrary and unconscious decision. It means that in the discovery phase of their research, they didn't see anything unique to the male stem cell, nor did they detect important differences in function between male and female cells. The result of not considering the sex of the stem cell was that their male mice died, and they didn't know why. So they thought maybe a postdoc made a mistake. If in doubt, blame the postdoc. But eventually, through a Gendered Innovations workshop in Norway, the team realized that they should consider also the sex of the stem cell. And they found that sex matching, so in their case, they found that sex matching male to male and female to female was the best combination, gave the best outcomes. But of course, you have to look at all the possibilities. You have to do the research before you can rule anything out. But then it's never that easy. In the case of stem cells, there are other factors that must be taken into account. One of the methods on our website that's very important is always looking at other factors that intersect with sex and gender. So for humans, it might be ethnicity, it might be SES, it might be other social conditions. But for cells here, it's important to look at other variables like hormonal status of the cell, the immunological or environmental factors. So it's not always so easy to tell the sex of the cell. Okay. In fact, the NIH, in this requirement that they put in place in 2016, do not require that you look at the sex of your cell when you're asking for funding. You have to look at the sex of the animal, everything else, but not the cell, because cell lines tend to degenerate over time. It's not really so clear sometimes what the sex of your cell is. But you can know the sex of your cell. You probably know this very nice article by Capel-Shaw, published in 2014, that provides a table, this is just a part of the table, of the most commonly used cell lines appearing in the American Journal of Physiology, Cell. And I have heard now that this journal sends back articles to any author who does not say what the sex of their cell is. So they will not publish now unless you say the sex of your cell. So let me interject here. I was talking about stem cells and introducing those into mice, and that sex matching was the best system there. Sex matching can be important for stem cell research, but it's also important for human organ transplant. For heart transplant, for instance, sex matching yields the best 15-year outcomes. And this is also true for kidney transplant, but lungs don't follow that pattern. So again, you can't make any assumptions. You always have to do the research. But now, even though we know the science and we know what sexing combination works the best, there's a gender problem for human transplant. And that is that women donate more organs. So even though a surgeon may know the best science, the surgeon has a man who desperately needs a heart, but men don't donate hearts as often. He only has a supply of female hearts. So we have to do something about the sex, getting the sex right in the science, and also understand gender issues in the society. Okay, so far, I've been talking only about analyzing sex, and now I want to open the discussion to gender. And if my clicker worked, I would have more time. So I want to just show you this image from Vera Ragitza-Grosek, who I think is well known to you here in Berlin. She's one of the powerhouses of gender medicine in Europe. And she has produced this very nice image showing the complex interdependency of sex. And so I'm sure you can still hear me. So it's very important to realize that human beings are a combination of sex and gender. So across the life course. So we all have genes and hormones, and this is the sex So sex influences health by modifying behavior, and gender behaviors interact with and influence biology. That's why we care. Gender behaviors can modify biological factors. For example, exercise strengthens bones, as we know, or social discrimination can lead to stress, which in turn impacts health. And these are social factors that differ by gender. So in our research, we have to look not only at sex, but also gender. I really don't understand. Okay, thank you. Okay. Oh, I'm supposed to click it stronger.
And you have a laser pointer over there.
Yeah, I know, but I like to walk around. It's, you know, right. Well, don't get me started on technology and gender issues. We're going to do a whole workshop on machine learning and gender issues and robots and gender issues, but that's a different lecture. So back here. So we have a potential danger, and I want to take you now to some animal research. I think I have time for one more example maybe. So authors in peer-reviewed journals often use the word gender when they mean sex. This is the #1 point in Lancet. We did an editorial in Lancet last December, and you have to use the word you mean. You can no longer get published by screwing up the meaning of the word sex and gender. So even in German, if you don't have separate words for that, you have to figure out if you're talking about biology or if you're talking about social issues. So because in animal research, we don't want to be talking about the sex differences, biological sex differences in animals, if the issue is really an environmental issue. And so here we have, these are the laboratory conditions within which the animal exists. And there are many types of conditions that influence a sex outcome in the research. Caging, for instance, is probably well known to you, diet, room temperature, but I want to give you an example from the researcher staff point of view down here. So we'll look at one quick example of how environmental factors relate to sex and how this might influence an animal experiment. And here the example is a social relationship between the experimenter and the animal. So this example is about the sex of the researcher. So it's an environmental condition, who is in the room, and the response of the animal. The example focuses on pain research. Importantly, experimenter sex may be a confounding variable in rodent research where stress is a significant factor. Zorg et al., a lab at McGill, was studying pain, and they induced pain in rats and mice. What they found was amazing. They found that rats and mice did not show their pain in the presence of a man, of a male researcher, as compared to an empty room, whereas the animals didn't care if a woman was in the room. So both male and female rodents had this response. And you know how these animal researchers, they have grimace charts. So pain, looking at pain in the lab is a really fine science. They look at the grimace on the, they have ways of measuring how the animal is showing pain. So this is called the male observer effect, and it can influence your research. And so what's going on? The animals smell male pheromones, and then they don't show their pain. According to Jeff Mogul, whose lab this came out of, this phenomenon throws into question all prior results from pain research. So in other words, we need to start over. So I guess I should stop. I wanted to talk about heart disease from a point of view of sex. Okay, I get to go on. So the next thing I want to talk about now is humans, and look at sex differences in humans for heart disease, and then gender differences. So we start to get the idea here. So heart disease research in women offers one of the most developed examples of gendered innovations, and it's amazing what we've learned over 20 years thanks to people like you. We know a lot about sex differences. For example, we know that coronary angiography, the gold standard of diagnosing patients with chest pain, is blind to women's pattern of disease. So you all know this. This is sex difference in heart disease and is now well understood. But let's move on to gender. And here, new tools are emerging. The question is, can we capture gender in variables that can be deployed quantitatively in research studies so that you can look at gender in clinical trials? That's the goal of this research. We at Stanford are attempting to develop such tools now, and we were inspired by a Canadian study published in 2015 led by Louise Pillett. I'm going to go through this study quickly because our tools are not ready yet. I see this Canadian study as extremely innovative in finding a way to reduce the complex issues of gender to measurable variables. So the Canadian team studied acute coronary syndrome and wanted to know why some patients have higher incidence of recurrence and death 12 months after diagnosis and what they can do about it. How can they intervene? To do this study, the PIs reduced sex to a variable. This is straightforward, so they looked at sex, and they found no variation in acute coronary syndrome between males and females. So they also started looking at gender. They first tested 17 variables and then found 7 factors that were significant, and from this developed a gender score. And I think these are very, very interesting. There are factors that very often are not considered in clinical trials. So they looked at the household primary earner, they looked at personal income, they looked at the number of hours per week spent doing housework. Did this have an impact on heart disease? As a professor who also had children, I think it does. They looked at the status of the primary person responsible for doing housework. They looked at the level of stress at home, and then they used the BIMS sex role inventory masculinity and femininity score. These, of course, they controlled for sex, age, the GRACE score, previous cardiovascular disease events, and a number of traditional risk factors for cardiovascular disease. So the results is that gender matters. Patients received a gender score of between 1 and 100, and patients with a higher femininity score were more likely to experience a recurrence of ACS regardless of whether they were a man or a woman. So a man with a high femininity score was more likely to suffer a recurrence of ACS, and a woman with a high masculinity score was not. I see this as innovative methodology. But I also see problems here, and this is why we at Stanford are redoing these tools. I think it doesn't make any sense to talk about men and femininity scores and women and masculinity scores. And we found, I mean, most importantly, these last 2 elements in this tool are not any more relevant. This BIMS sex role inventory. If you ever come across the BIMS sex role inventory, run in the opposite direction. So we at Stanford have, well, I should say that the BIMS sex role inventory was ironically developed at Stanford in 1974, and it reflects the gender attitudes and behaviors at that time, when Stanford was incredibly white, incredibly privileged, and incredibly athletic. Right? We get a lot of Olympic medals. So it really is not a kind of score that you can use here in Berlin, that anybody in the United States should use. So we at Stanford have launched a new study in collaboration with a Canadian group. Louise Pillett is a member of our study, and with European colleagues, entitled Gender Variables in Health Research. And it's been 2 years. This is really difficult to do, but we hope by the beginning of next year to have a patient questionnaire available that can be integrated into clinical trials. I will stop there. Thank you very much.